So first, thank you for this invitation. So indeed, I will talk today about EV engineering and especially numbers in EV engineering. So you will learn about yields, rates, and efficiencies. And I tried to use these numbers to highlight some of the practical limitations we can face when we work in EV engineering. So first, why would you engineer this already amazing EV? You may want to use them as drug delivery system. In this case, you may want to change their cargo to load a cargo of therapeutic interest. But you may also want to boost the properties of EV that already have a therapeutic interest, such as EV derived from MSC, as an example. In this case, you can want to work on their surface modification, as an example, to increase their biodistribution, to control their intracellular behavior, as an example, to increase their endosomal escape properties. And how you could engineer these EVs, you probably already seen such a scheme, but you have basically two main approaches. One, working with the cells that will produce the EVs. In this case, you can work on cell priming, passive loading, genetic engineering, especially with targeted genetic engineering, which is the most efficient till now. But you can also work on EV that are already isolated, so it's called then exogenous engineering. You may want to use some membrane permeabilization processes to allow your cargo to diffuse inside EV lumen. In this case, you will have to use huge concentration of your cargo, so it could be a problem. We can discuss about that later. You may also want to fuse these EVs with synthetic nanoparticles that will carry some of your cargo of interest or a surface molecule of interest to try to form hybrid particle. And for surface modification, you can act either on lipid anchorage or on chemical modification of the proteins that are present at the EV surface. So, first, before starting with the numbers, I just want to be sure that we are on the same line. So I will take a few minutes to talk about terminology, less than a minute. So I will use pharmaceutical engineering terms adapted to the EV field. So when I will talk about EV engineering efficiency, it's to answer the question, how many EV are engineered? So it's the number of engineered EV regarding the total EV that you have in your suspension. Here, as an example, it will be 25%. The next interesting thing is the engineering rates. It's how many molecules per EV I can load. So here it's, as an example, 18 molecules per EV. And the last terminology, the last parameter that I will talk about is the encapsulation yield. There it's more focused on the exogenous loading. It's the amount of cargo or molecule of interest you will bring at the beginning of the process and the amount that will be actually associated to your EV after all the process and purification steps. So as an example here, it will be 6%. So now, let's start with examples. So I will first talk about endogenous engineering, for protein loading. And this example by Silva et al., published in 2021, showed that active loading of GFP by genetic fusion of a protein of interest, here it's GFP, to scaffold proteins is highly important. Indeed, by comparing the loading efficiency, they've shown that depending on your scaffold protein that is enriched in EV, you can hope to have around 30 to 40% of EV that are actually loaded. By contrast to known or nearly known when you have no active targeting, or low amounts when you have some of the less efficient proteins. So here, CD63 was the most efficient, but till then, Samir El-Andalousi group shown that tetraspanin 2 and 3 can outperform CD63. It really depends on your system. In this study, they also shown that the loading rate can vary a lot depending on the scaffold protein. It can go from 9 to 173 GFP per EV, also depending on the scaffold protein. They also highlight that there is an heterogeneous distribution of this loading among EV subpopulation. So this highlights that the active targeting in the endogenous engineering is really crucial. The selection of your protein, of your scaffold protein is crucial, and you can expect an average of 30 to 40% of EV that will be loaded with a protein scaffold, protein of your protein of interest. Sorry. Then what about nucleic acids? With nucleic acids, it's even lower in terms of loading rates. I just highlight here 2 studies, because of time, who shown some interesting loading rates. One with siRNA loading by the Reschke, published in 2020 by Reschke et al., using the backbone of Premier 451, who was described as being enriched in EV. They managed to load 1 to 30 siRNA per EV. It's quite a lot in the field. Of course, it's a bulk characterization. It's evaluated by qPCR. But more recently, looking now at messenger RNA loading, this recent study by Dong et al., who combined genetic engineering using an RNA binding protein domain that was fused to CD64, enriched in EV, and loading mRNA with nanoelectroporation, they reach an efficiency of 70%. So it means that 70% of the EV were actually loaded with mRNA. It's quite a huge amount. And it's one of the first studies who really reported engineering efficiency for nucleic acid loading. And they managed to have 1 to 2 mRNA per EV. So just to sum up quickly on endogenous engineering, the main advantages of endogenous engineering is that the cells are doing the job, and it's an interesting strategy, mainly to load biomolecules. But the limitation of this approach is that these sometimes complex genetic engineering processes are appropriate for highly transfectable cells, but maybe less sometimes for primary cells, such as stem cells. Then the fusion with endogenous proteins, we heard about that already, could negatively affect EV production and EV content, so it has to be checked. So now switching to exogenous engineering and using this time small molecules loading, I wanted to highlight this study by Yan group, who work on doxorubicin loading using active processes into milk-derived EV. And this, among the interesting thing in this article, I highlight this fact that starting suspension quality really matters, because what they shown, working here with nano FCM analysis of the engineering efficiency, of the loading efficiency, they showed that with doxorubicin, they were able to load doxorubicin in 20% of milk-derived particles. But they also figure out that in this milk-derived particle, they have nearly 50% of particles that were actually not lipid vesicles. There was casein nanoparticle, proteins. And when they focused on lipid vesicles with lipophilic dye, they shown that this time the percentage of doxorubicin positive particle inside these lipid vesicles was 60%. So this highlights that it's really important to avoid the presence of contaminants in the starting solution of EV, to well know your starting solution. It's not just to be boring, it's just because you can have more interesting results and more reproducible loading efficiencies. And the same observation was done with curcumin, or cholesterol-anchored molecules, in the recent work published by Tretton et al. In 2023. And to continue with this example, so this study with curcumin loading, one of the interesting thing they shown, working with different concentration of curcumin that they wanted to associate to EV. They shown that when high concentration of curcumin were used, you can have by cryotane imaging, you can see that there is large, dense electron particles. So here they've shown that you can have an hypersaturation of the EV surface. Of course, it will impact cell interactions. And this highlights that it's particularly important to think well at your cargo concentration. So in this case, you have an hypersaturation at the EV surface, but you can also simply have aggregates aside your EV that can be confused with EV because it can have the same size. It was published 10 years ago with siRNA precipitation that was observed after electroporation. And in our lab, we also worked with protein loading, and we shown that protein can also easily form aggregates that can be confused with EVs. So it's particularly important, I think, to try to use free cargo control when you are doing EV engineering or EV loading Finally, still to continue on this work by Tretton et al., they looked at the loading efficiency with curcumin then, and they showed that 25% of EV were loaded with this curcumin. They also study the interest of the lipid anchorage strategy. And here they show that this time 90% of their particles were loaded with this lipid anchorage strategy. So what they highlight is that for hydrophobic compound loading, you can have low loading efficiency. It's often heterogeneous. And the lipid anchorage seemed to be more interesting in this case. It was also published by Samir El Andalusi group with another lipid anchor that you can have really interesting engineering efficiency with this strategy. The limitation is that you will have your cargo exposed at the surface, so it can be a problem if you work with a fragile cargo. Finally, the last advantages of this strategy is that all the vesicles have lipids, so it's not restricted to a specific subpopulation based on the protein composition. And, So in our lab, we are also using lipid anchorage, but to associate polymers, because we are really interested in the connection between polymers and EVs. And we've shown that working with polyethylene glycol alternative, polyoxazoline, we were able to enhance plasma stability of EV after intravenous injection, and above all, we shown that we were able to increase the tumor accumulation. And it's interesting because we compared it to the gold standard polyethylene glycol, and we shown that there was really different biodistribution, especially tumor accumulation, while using polyoxazoline. So it's an interesting alternative, and we also shown that we had a decrease in liver accumulation. We are also using lipid anchorage to associate other cargoes to allow intracellular delivery, but I have no time to talk about that, don't worry. So if you're interested with the connection between EV and polymer, I invite you to read our recent review, because polymer can be interesting at each step of EV life, in production, detection, and also, of course, therapeutic application, as you can see here, with association to different type of biomaterials. Finally, one of the challenges, it may be self-centered on the horizon, but one of our main challenge in the lab is to try to understand precisely how these lipopolymers could organize into or onto EV membranes. But it can be also the case, it could be also interesting in general point of view to try to characterize and know what happened when we engineer EV membrane. In the case of polymer, we know a lot about how polymer can insert in simple membranes. You can, depending on the density, on the molecular weight, you can have different conformation. You can have brush conformation, as you can see here, mushrooms. But we don't know a lot about how this polymer can organize at EV surface, how they can be retained, and what can be the impact on the corona and so on. There is a lot of things to do in this topic. And so finally, to sum up on exogenous engineering, again briefly. So exogenous engineering have the advantages that there is no genetic manipulation. Theoretically, it's applicable to all EV types, and it could allow for greater control of the cargo dose if the dose is adapted at the beginning of the process. The limitation is that the efficiency will really depend on the starting suspension. It's important to well know your starting suspension. It can really impact the loading efficiencies and the reproducibility. The permeabilization processes, I didn't have time to talk about that, but it can really impact EV integrity and cargo behavior. So, of course, it has to be checked. And finally, it's important. There is a lot of work to do on the purification step because you have to adapt your purification protocol to the protein, to the cargo you want to load. You won't do the same if you want to discard protein or remove small molecules or nucleic acids. And it's really important to check, to well choose the purification protocol. Finally, some take-home messages, which from my point of view are important. I think you understand that the starting suspension of EV is really important to look at what's in the tube at the beginning and at the end. It's really important looking at EVs, but also on cargo or molecule you want to associate. I hope that you're convinced that numbers matter. It can give interesting information. So it's important to characterize this engineering and report the engineering efficiency, the loading rate, the loading ratio, with using bulk characterization, but also single EV characterization, as I shown. It's particularly important, and it can give interesting characteristic. The localization also is important. Where is your cargo, at the surface or in the lumen? And to finish, I wanted to say that if you want to move to a therapeutic product, I think that one of the most important things is to try to keep it simple, because to go to the clinic, you will have to keep it simple. And if you have to modify your EV too much, or if you have to work too hard on improving your EV ability, it's maybe that EVs are not the right choice. I don't want to be pessimistic, but they are not the answer to everything. So sometimes it could be interesting to challenge EV with synthetic nanoparticles, which can perfectly do the job. Just ask the question. Often when you challenge EV with synthetic nanocarriers, they generally win, but not all the time. So just ask yourself this question. And with that, I'm done. I'm sorry because I think I'm late. I want to thank you for your attention, and I'll be pleased to discuss with you about EV engineering.